Hello, I'm going to explain you the blue white selection, and this explanation is for lac jet complementation, that is alpha complementation also. This is the vector which contains the antibiotic resistant gene. It has got the replication origin and lac jed gene. We are going to focus on this lac jed gene. You can see that there are multiple cloning site. This lac jed gene contains the multiple cloning site, like uh, it has got the recognition sequence for PST1, EcoRI, EcoRV, etc. Means whenever we will cut this plasmid with any one of the restriction endonuclease, it is going to cut this plasmid in this lac jet. Okay, now let's see next. This, as we know, as you have already studied lacoperon in SYBSC, this is the lacoperon having Z, Y, and A gene. Z codes for beta galactosidase. Whenever it undergo transcription, mRNA will be produced. This is the mRNA, and it will undergo translation, and there will be beta galactosidase production. And we know it that beta galactosidase, it acts on lactose and it get converted into glucose and galactose. Okay, this is just the information regarding the lacoperon, because we are going to focus on this Z gene. This lac Z gene is divided into two parts. The initial part is known as the lac Z gene, whereas the latter part is known as the lac Z B prime. Okay, lac Z alpha part, it gives rise to mRNA which produce the alpha fragment, whereas lac Z beta prime, it undergo transcription, produces mRNA and gives rise to beta fragment. So this explain you that the complete Z, what we say, the complete beta galactosidase is made up of alpha fragment and beta fragment. Alpha fragment is a part of this beta fragment, means when alpha will combine with beta, you will get the active beta galactosidase enzyme. Now, this is, as I explain you, this is the alpha fragment of LAGZ gene, this is the beta fragment of LAGZ gene, and whenever alpha will unite with beta, you will get the complete beta galactosidase, that is the active enzyme, and it is capable of carrying out hydrolysis of lactose. In addition to lactose, it acts on X-gal also. X-gal is, it's another substrates where beta galactosidase act, and it it produces a blue colored compound. Now we are going to use this property of beta galactosidase, that is it act on X-gal and it break the galactoside bond and it gives rise to formation of blue color. As we have started with the blue white selection, now this is again the very first diagram which I explain you, that this is the vector having lac Z gene containing multiple cloning site. Okay, so you can use any one of those restriction endonuclease which are mentioned over here to cut this vector, and you can insert the gene which you want to clone. Now this, you will get the recombinant plasmid, because here we are going to insert the gene of our interest. Okay, now this recombinant plasmid is transformed into the bacterial cell, and these bacterial cell in which the transformation has been carried out are allowed to grow on a medium containing antibiotic for selection. Now how we are going to select uh, these transformant? You can see that there is a gene which codes for the antibiotic resistance, like here it is specifically mentioned that it is ampicillin resistant gene. So you add minimum inhibitory concentration of ampicillin in that medium, so that only the cell which have received this plasmid will be able to grow, because now these cells will show the uh, antibiotic resistant gene, that is resistance towards ampicillin. In addition to this, you add IPTG in that medium. Now what is the function of IPTG? This IPTG will help to induce the expression of LAG gene. And another thing is you add X gal in that medium. Now what is the role of X gal? Whatever the beta galactoside is which will be produced, that beta galactoside will act on X gal, and it will produce, as I mentioned in previous slide, it will act on X gal, and the blue color product will be formed. Okay, and this blue color will give which will helpful to distinguish between the blue and white color colony. Yes, there are two possibilities. Either your foreign gene will get inserted in LAGZ. Yes, this is the cutting site. There are two possibility. Either your foreign gene will get inserted in this vector, or it will not get inserted, right? So let's see one by one that what will happen if the foreign DNA get inserted or if it will not get inserted. First, in first possibility, let's say that if this vector, plasmid vector, will undergo transformation without carrying foreign DNA. In that case, what will happen? This lac Z will be intact. It will not get disturbed, and there will be production of alpha fragment, right? Because foreign gene is not inserted. And the another beta fragment will be there from the E. coli chromosome. E. coli chromosome will produce the beta fragment, and where your plasmid will produce the alpha fragment, and there will be fusion between alpha and beta. You can see over here, this is the alpha fragment which is produced from our vector, and this is the beta fragment which is produced from E. coli chromosome, and there will be alpha complementation between these two fragment, and active beta galactosides is there, and these beta galactosides will act on X gal which is already there in the medium, and you will get the blue color colony. Okay, next possibility is transformants with recombinant DNA. It means that when foreign DNA, foreign gene get inserted in this plasmid, that time what will happen? This lag Z will get disturbed, and as it is not intact, so it will not produce alpha fragment, right? But beta will be there from E. coli chromosome. But in absence of alpha fragment, there will not be production of, oh, I'm sorry. Yes. There will not be production of active beta galactosidase, and this beta galactosidase will not act on X gal, and therefore there will not be formation of blue colored colonies. So in this case you will get the white color. So it is very easy to distinguish that if you get blue color colony, it means that foreign gene is not inserted, and if you get white color colony, it means that foreign gene is inserted, and whatever the genetic engineering steps which we have performed, those steps were correct. Yes, you can see this kind of blue and white colony. These blue colony, it means that foreign gene is not inserted, and white colony means foreign gene is inserted. This is the way we can distinguish between the two clones, or this is nothing but the cell in which foreign gene is inserted properly, and this is the colony in which foreign DNA is not inserted. So this is called as the blue white assay, or whenever there will be question on alpha complementation, you need to write all these steps. Thank you.